Think about the last time you got a small cut. You probably cleaned it, maybe put a bandage on, and then forgot about it. That simple act of forgetting is a luxury, a modern miracle built on a silent, invisible protector: antibiotics. For almost a century, we've lived in a bubble of security, where a bacterial infection was an inconvenience, not a death sentence. But what if that bubble is popping? Silently, all around us, in hospitals, on farms, and inside our own bodies, a revolution is happening. The microbes are learning. We tend to think of evolution as a slow, vertical process. Parents pass genes to their children over generations. But bacteria don't just play by those rules. They have a secret, ancient network, a kind of biological internet that has existed for billions of years. They can share genetic information horizontally with their neighbors, even with entirely different species. A harmless E. coli in your gut could receive a software update from a dangerous Klebsiella pneumonia it just bumped into. This isn't just inheritance, it's a peer-to-peer file-sharing network for survival skills. How does this sharing actually work? One method is called conjugation. It's the closest thing bacteria have to sex, where one bacterium extends a tiny tube, a pilus, and directly transfers a packet of genetic code to another. Another way is transformation. When a bacterium dies, its DNA can burst out into the environment. A nearby bacterium can then just absorb these free-floating genetic fragments, like scavenging for useful spare parts. And then there's transduction, which is perhaps the strangest. A virus, a bacteriophage, can accidentally pick up DNA from one bacterium it infects and then inject that DNA into the next bacterium it attacks, acting like a microscopic biological syringe. The information packets being shared are often plasmids. These are small, circular loops of DNA separate from the bacterium's main chromosome. Think of them as genetic USB drives. They can be copied, passed around, and they often carry the most potent survival tools, including resistance genes. What's truly alarming is that a single plasmid can carry the code to resist multiple different antibiotics at once. So when a bacterium gets one of these plasmids, it doesn't just learn to defeat penicillin. It might simultaneously learn to defeat tetracycline, erythromycin, and a half dozen others. It becomes a superbug in a single transaction. So what does a world where this process runs rampant actually look like? It's not necessarily a dramatic apocalypse. It's a quiet, slow-motion crumbling of modern medicine. An organ transplant, which relies on suppressing the immune system and fighting off infection, becomes almost impossible. Chemotherapy, which decimates a patient's white blood cells, becomes a lethal gamble. A routine C-section, a hip replacement, even a visit to the dentist could introduce an unkillable infection. The future without effective antibiotics looks a lot like the past, where a simple scratch from a rose thorn in the garden could lead to a fatal case of sepsis. We are in an arms race against an opponent that doesn't sleep and doesn't follow our rules. It takes us, on average, 10 years and over $1 billion to develop one new antibiotic. A single bacterium, like E. coli, can replicate in as little as 20 minutes. That means in the 10 years it takes us to create one weapon, they have gone through over 260,000 generations. That's equivalent to about 5 million years of human evolution. Every single one of those replications is a chance for a random mutation. Every interaction is a chance to acquire a new plasmid. We are fighting a hyper-adaptive, globally connected superorganism that learns faster than we do. So, can we win? Maybe winning is the wrong word. But we are developing new strategies. One of the most promising is phage therapy. We can use those same bacteriophage viruses that spread resistance genes, but this time we select phages that are natural predators of specific superbugs. It's like programming a microscopic hunter-killer drone. Other researchers are using CRISPR gene editing technology not to edit human DNA, but to create tools that can enter bacteria and precisely snip out the resistance genes from their plasmids, effectively disarming them and making them vulnerable to old antibiotics again. We're even learning to disrupt their communication, using molecules that block quorum sensing, preventing them from coordinating their attacks. These new tools are ingenious. They are a testament to human creativity. But they also reveal a deeper truth. We are not fighting an invader. We are fighting life itself, in its most ancient, adaptable, and successful form. Bacteria have been on this planet for at least 3.5 billion years. They have survived planetary freezes, asteroid impacts, and atmospheric transformations. Our entire age of antibiotics has been, on their timescale, less than a blink of an eye. The question isn't just whether we can invent the next clever weapon. It's whether we can fundamentally change our relationship with the microbial world, recognizing that we are not its masters, but merely its most recent and perhaps most fragile inhabitants.
Monotony has grown fast. Nearly 2,700 people have watched in the last few weeks, yet less than 0.03% are subscribed. It would mean a lot if you joined by hitting subscribe. And again, thanks for watching.